Hello everyone, my name is Saengkyongam. Today I'm going to present our work on learning joint nonlinear effects from single variable interventions in the presence of hidden confounders. This is the work that I did with Ricardo Silva at the University College London. I'll start by giving an example that will illustrate the motivation of our work. Imagine that we want to study potential drug interactions in kidney disease patients. And let's say we have the blue drug that directly controls sodium level, which in turn has an effect on the kidneys, and the yellow drug that controls blood pressure, which also then has an effect on the kidneys. Also imagine that we are given these underlying causal relationships. However, in reality, we may not be able to observe all the confounders, and that we may have hidden confounders between each pair of the variables, like this. The aim is to understand the joint effect of these two drugs on the kidneys. So essentially in this example, we have two control variables, x1 and x2, and the outcome variable y. And here's the target regime that we want to study, where we intervene on x1 and x2 simultaneously. If we can actually obtain data from this regime, then the joint effect can be estimated by just standard statistical methods. However, in reality, we may not be able to perform randomized experiments where we randomly prescribe these two drugs to the patients. We then need to rely on other data sources to estimate the joint effect. The common data source is the observational data, but in this observational regime, the joint effect is not identifiable because of hidden confounders. Therefore, we need to rely on additional data sources. And in this work, we assume to have access to data from regime B and C, where we intervene on each of the control variables separately. In our example, we may have data from previous randomized control trials on each of the drugs. The question is then, how could we combine the data from regime A, B, and C to make prediction in the target regime D? We are now ready to formally define our problem definition. In our work, we consider k control variables and one outcome variable. We represent the data generating process by the following SEM. Essentially, we say that the outcome variable causally depends on other control variables, and there is a certain causal ordering among the control variables. Also importantly, we do not assume that the noise variables are jointly independent. In other words, we allow for hidden confounders. We are given two sets of samples, one from the observational regime, where we don't perform any intervention, and another set from the single variable interventional regime, where we intervene on each of the control variables separately. The goal is to estimate the effect of the joint interventions, which is the expectation of the outcome, given that we intervene on each of the control variables simultaneously. So now the next natural question to ask is whether the effect of the joint interventions is identifiable from our given data sources or not. The answer is no if we don't assume any structural assumptions. And we can see this using the following causal example. We construct these two SEMs, and where we show that the two SEM entail identical observational distribution and also entail the same single variable interventional distribution, but they use different joint interventional distribution. And by this example, the effect of the joint intervention is not identifiable without further structural assumption. Then how do we proceed? The solution is to add some restrictions to the underlying causal models. In particular, we chose to use the additive noise models, where the noise variable only affects the endogenous variable in an additive way. We then have this following underlying SEM. We also assume that the noise distribution is a multivariate Gaussian, and again the noise can be correlated, which allow for hidden confounders. And here's our main theoretical result we provided. We basically show that the joint interventional effect is unidentifiable under additive Gaussian noise model. And we use the following main assumptions to provide identifiability. You can see our proof in the main paper. So now we have shown the identifiability of the joint effects. The next question to ask is how to estimate it from as finite samples. We propose a simple estimation procedure by pooling all the data from different regimes and jointly maximizing the combined likelihood. And because of the additive Gaussian noise and acyclicity assumptions, we can then define the likelihood for each of the regime as follows. We can then combine the likelihood to obtain the final likelihood. And then we can alternately solve for the model parameters theta and the noise covariance matrix sigma. And lastly, we conduct several experiments to validate and compare our approach to a baseline. The first experiment we conducted was a synthetic experiment where we aim to verify the identifiability result. In this setup, we have 3 control variables with hidden confounders. We also have a baseline, which is a direct regression approach, in which we pull all the data and directly regress y on x. And here's the result where we show the consistency of our approach. The blue curve is from our method, and the yellow curve is from the baseline. We also performed the experiments on both linear and nonlinear underlying functions. As you can see from the plot, the mean absolute error of our method decreases and approaches to 0 as sample size increases. And this indicates the consistency of our method, while the direct regression clearly suffers from the bias because of hidden confounders. We then perform a more realistic experiment where we obtain the data from a realistic underlying data generating process. We first learn the underlying SEM from the Dreamfold gene expression dataset, which contains 10 variables. We fit the model using the observational data and the ground truth causal structure. We then select the last node according to the causal ordering as our target variable and the rest as the control variables. We also emulate hidden confounding effect by imposing the correlation between the noise variables. The training and the test data are then sampled from the underlying SEMs. And here's our result. The left plot shows the performance of our method as we increase sample size. It is clear that our method performs much better than the baseline, and approach to the oracle performance, which is the model that used the underlying parameters. On the right plot, we illustrate the effect of hidden confounding, where we measure the performance of our method as we vary the hidden confounding level. We then, when we have a small hidden confounding effect, as expected, the performance of our method and the baseline is comparatively the same. However, as we increases the confounding level, the baseline gets worse and worse performance, but our method is unaffected. This shows that our method is able to disentangle the hidden effect. Is able to disentangle the hidden confounding effects. And that's all I have. Thank you so much for your attention.